In this section, we will go through the normal cell-to-cell communication modes as well as the types of cytokines receptors. Normal cell-to-cell communication depends on chemical messengers and receptors on cell surfaces that can recognize those receptors. The steps involved in this are signal perception or reception, signal transduction, and cellular response. Signal perception means binding and recognition of the chemical messenger by the receptors on the cell membrane. So in this step, a signal is received by cell. It happens when a ligand or a signaling molecule binds to a receptor on the surface or inside the cell. As you can see here, a ligand binds to the external receptor of a cell. 2. The second stage is transduction. When the signaling molecule binds to the receptor, it changes the receptor protein. This change in the receptor sets off a series of events, like the domino effect. Each relay molecule or the second messenger in the signal transduction pathway changes the next molecule in the pathway. The final protein that was activated in the previous stage triggers a specific cellular response. Regulation of gene expression, opening or closing an ion channel in the plasma membrane, and promoting a change in cell metabolism are some of the common outcomes of cell signaling. Since we know that the cytokines act as messenger molecules and form a network of communication between the cells, it goes without saying that the cells must connect with one another to transmit signals. Cells of the immune system communicate through the exchange of secreted cytokines. Depending on the spatial location and identity of the cytokine-consuming cell, such communication can be autocrine, signaling to self, paracrine, signaling to neighbor cells, or endocrine, signaling globally through circulation. Let's go through them one by one. Long-distance endocrine signaling involves the production of signals by specialized cells and their release into circulation, which then transports them to target cells throughout the body. When it comes to cell communication through cytokines, an immune cell is stimulated, either by the presence of a pathogen or other cytokine in the circulation, and more cytokines are released by the said immune cell. These cytokines travel through the bloodstream throughout the body and perform their various functions. This type of signaling is more common in the endocrine system. Hormones are signals that the body produces in one area and sends through the bloodstream to distant locations. Another type of cell-to-cell communication method is paracrine signaling. Only cells in close proximity to one another are allowed to communicate using this way. The two adjacent cells release cytokines or other messenger chemicals. The receptors on the target cell then detect them. Often cells that are near one another communicate through the release of chemical messengers, ligands that can diffuse through the space between the cells. This type of signaling, in which cells communicate over relatively short distances, is known as paracrine signaling. The third way to cytokines communicates with other cells is called the autocrine signaling. This type of signaling involves the production and secretion of an extracellular mediator by a cell, followed by the binding of that mediator to receptors on the same cell to initiate signal transduction. Just like how I mentioned that endocrine signaling is more commonly seen when it comes to hormone signaling, in the immune system the paracrine and autocrine modes of communication is more commonly seen. After talking about the different forms of communication, let's move on to the topic of how cytokines exercise their functions. Specific cytokine genes are activated in response to an initiating or stimulating signal. The target cell would ultimately undergo cytokine-mediated signaling as a result, and this would have a number of biological effects on the cell. It could either divide, differentiate, or occasionally even go through apoptosis. So cytokine signaling could lead to a range of outcomes. The cytokine signal is received by the receptor. Cytokine receptors are also often only expressed transiently. For example, the complete IL-2 receptor is only transiently expressed by activated T cells. Again, this mechanism has evolved to prevent inappropriate activation of the immune system. The three main types of receptors are the common cytokine receptor family, the Chemokine receptor, the TNF receptor. There are many other types of receptors, but we will only discuss the three main types here. Most cytokines use the common receptor molecules, sometimes called the hematopoietin receptors. The receptor can be in different shapes and sizes. Cytokines act as growth factors and interferons to these receptors. The majority of common cytokine receptors are not highly expressed in fully resting cells. Rather, they are more frequently increased after a cell has been activated, such as when a T cell has been stimulated via its T cell receptor. When cytokine binds to its receptor, it causes aggregation of the receptors at the cell surface. A cytokine-mediated cell activation pathway known as the JAK-STAT pathway has already been discussed in detail in previous videos. Let's just revise it briefly. JAKs remain inactive when they are spread under the cell's surface. The JAK enzymes become activated when the receptors are brought together by cytokine binding, and then they phosphorylate one of several transcription factors, called signal transducers and activators of transcription, STATs. The activation of this pathway is not possible without the presence of the common cytokine receptors. Janus kinases, JAKs, are normally loosely associated with the cytoplasmic portion of the cytokine receptors. Once STAT molecules have become phosphorylated, they form a dimer, migrate to the nucleus, and activate the transcription of specific genes. Chemokine family of cytokines that play an important role in relation to attracting cells into inflamed tissues. They also have a role in leukocyte homing. For example, chemokines secreted by cells in lymph nodes attract B cells to germinal centers and dendritic cells. They also attract T cells to the T cell areas. They play an important role in carrying out the immune response, so the presence of receptors that can bind the chemokines is also important. Many chemokines can bind multiple receptors, and the majority of receptors can bind multiple chemokines. Lastly, we have the TNF receptors. TNF uses a special family of receptors. TNF, or the tumor necrosis factor, is secreted and can be active as a membrane-bound form. It can also be cleaved from the secreting cell membrane and is then free to cause local or remote effects. Both cell-bound and cell-free TNF are present as a trimer and bind to one of two types of receptors, which are also trimers. The cytokines in the TNF family are either soluble or membrane-bound. They are involved in immune system development, effector function, and homeostasis. The effects of TNF depend on the target cell it binds to. When TNF binds to infected cells, it induces apoptosis. On the other hand, when TNF binds macrophages or endothelial cells, it induces the transcription of genes. That is all about the main receptor types to which the cytokines bind and bring about different changes in the cell. You can deduce from this knowledge that without the presence of these receptors, it is not possible for the cytokines to initiate these important cellular responses that play a role in maintaining the immunity of the individual.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.